Right, hello Year 13. Today I've decided to do a screencast for this so that you can take your time to work through it. There are a couple of practical techniques that we need to go over. Today I'm going to cover two of them. The first is microscope calibration using your eyepiece graticule and stage micrometer. So at the end of the day, you have to use these two terms in any of your answers with anything to do with taking measurements down a microscope or using a microscope. Before we get into it, you must be confident with moving between units, okay? So you must know that in 1 millimeter, there are 1,000 micrometers, okay? A micrometer being this symbol here, okay? There are then 1,000 nanometers in a micrometer. However, we talk about that a lot less with stage micrometers and eyepiece graticules because light microscopes cannot magnify to this degree. We can only get to here due to the magnification limitations of light microscopes. So, our eyepiece graticule, you may recall, is an actual eyepiece. Remember that we'd slot into our microscope in place of the ×10 eyepiece, the normal one, and on it will have a scale, a visible scale, okay, that's marked on there. The distance between the lines on this scale will change as the magnification changes. That's why we need to calibrate each magnification so that we know when we are looking at our object on the stage, we know that at that magnification, when we're using the scale on the eyepiece graticule, if we measure across and count the number of eyepiece units, we know what that equates to. Now, in order to calibrate your eyepiece graticule, you use a stage micrometer. This is a glass slide with a 1 millimeter measurement marked out, and that millimeter is subdivided into 100 sections. Okay, therefore, we can say that each of those sections, those segments, is going to be equal to 10 micrometers. Okay, because if you remember, 1 millimeter is equivalent to 1,000 micrometers. Okay, so if we divide that millimeter up into 100 segments, yeah, we need to get from that 100 to 1,000. We times it by 10, so each one is worth 10. Okay, micrometers. Okie dokie. So your eyepiece graticule will appear like this. And you'll look through it, and your image will appear underneath it. What does that say? Yeah, okay, fine. We've talked about that. Yep. And again, if you were doing this in the lab, you'd turn it to you always, when you end up with two scales, because you'll have the stage micrometer underneath it, you can always determine which one is the eyepiece graticule by rotating the eyepiece. Okay, so on low power, okay, we would end up with a situation like this, where we are looking through our eyepiece graticule here, which remains the same size, in our field of view on every magnification. And then our stage micrometer scale will be here. And what we would do is we would line up, okay, our stage micrometer scale with our eyepiece graticule scale. Yeah. Now, when you look down the microscope, you would then count how many eyepiece units are equivalent to, in this case, all 100 stage micrometer units, or the total 1 millimeter. So here we would go back, and you would go, it's equivalent to 4, but I would remind you that actually each one of these is divided into 10 segments, so it's actually equal to 40 eyepiece units. Okay, so the total stage micrometer scale, the 1 millimeter or 1,000 micrometers, which they're the same, is equal to 40 eyepiece units. Therefore, we are dividing, okay, our 1,000 micrometers into 40 eyepiece units. So we divide 1,000 by 40. Each one of those eyepiece units will be equivalent to 25 micrometers. And again, you would have done this at the time. Some of you would have got it, some of you wouldn't. You are not going to have to calibrate a microscope in the exam. However, you might have to explain it in answer to a question. So, how do we explain it? I would encourage you to learn something like these 3 sentences. Okay? First of all, you have to mention both pieces of equipment. Yes? So you do that in your first sentence. You will need an eyepiece graticule and a stage micrometer. But if you can say to the examiner, you know where they go. You are inserting the eyepiece graticule. That tells the examiner what you're talking about, and you place the stage micrometer onto the stage of the microscope. The examiner is going to be very impressed. They're going to go, okay, this guy knows exactly what they're talking about. And then line up the 2 scales. You could talk about bring them into focus. That's fine. Then you have to talk about it, and it's effectively just saying you will count how many eyepiece units are equal to a set number of stage micrometer units, and then you divide the stage micrometer length. Okay, so you in millimeters, or I would encourage you to say in micrometers, divide that length by the number of eyepiece units it is, and that will give you, to find the length of 1 eyepiece unit. So it's not hard, okay? I know, it was difficult. We often start off with it in year 12, and it's sort of it's to try and show you guys that you're not doing GCSE biology. However, you shouldn't have too much trouble learning this technique and how to answer it, an exam question on it in terms of a method. Okay, the next one, serial dilutions. So again, you did this on several occasions in your PAGs. If they're asking for a method, if they're asking for you to explain how you would, create a set of concentrations, they may well not tell you the term. They'll ask you what process would you use, and you'd get a mark for saying you would do it by serial dilution. And then it might say, then explain it. And again, you've done this in block tests, in the past. They will always ask you how to create. Oh, sorry, that should say distilled water. How to create your new set of concentrations by dividing it in half. Okay, they're not looking for anything more complicated than that. It will never be anything more complicated than that, unless it's talking about calculating bacterial populations. But in that case, they won't get you to do the calculate the actual serial dilution. Okay, they'll give you that. So here, you simply halve the previous sample and then add the same volume of distilled water. Yeah. So when we look at it, if we were given a solution, a starting solution of 1.5 molar sucrose solution. Yeah, so this was our potato osmosis, put the chips in, and measure the mass and all of that. Here, your starting solution, you'll notice you always start with double the volume you need. Okay. Well, actually, no, that's not true in this one. Sorry. You should always start in your answer. You should always say you start with double the volume you need. So in this case, we're going to take 20 cm cubed here out of your 1.5 molar sucrose solution, and you are going to add it to 20 cm cubed of distilled water. So you will end up with 40 cm cubed here, okay, and of 0.75. And once you've mixed that around, you can then take 20 cm cubed of your 0.75, add it to 20 cm cubed of distilled water to get your 0.38, okay, and so on and so forth, okay, all the way down. Now, here, what they're saying here is, actually, they are trying to indicate your 1.5 molar sucrose solution, they've made it by presumably taking a 3 molar solution and adding to 40 cm cubed of distilled water. What they're trying to say is you would end up with you start with double the amount, so that if you need 20, you need to be left with 20 for your test, for your samples, you're left with 20 here. Double, you make 40, then you take 20 out, you're left with 20 here. You get 40, take 20 out, left with 20 here, the desired volume. So, like I've said here, answering an exam question, you start, you take your starting concentration and measure out double the amount that you will need. Okay, so if they say you need to create a series with 50 cm cubed in each, or 25, here you go, 25 mil, 25 cm cubed, then you start with double that, 50. Okay, you measure out half of this and add to the next boiling tube, and then measure out an equal volume of distilled water to halve the concentration. And your simple serial dilution method will always be this. It will always be halving it. Okay? Like it says there, you'll end up with double the volume of the final concentration, and that is expected. That is the procedure. You cannot do this any other way. Okay? You can't take out, do a calculation and take out 28 and add 12, to get your desired concentration. That's not the serial dilution method. This is the serial dilution method. You start with a concentration and you will always be able to halve it to get to each one. Okay. Now, we talked about bacterial colonies, okay, and serial dilution. And what we're going to do is talk through a question, okay, an exam question, and have a look, excuse me, at how that pans out. I will have shared this presentation with you as well as this video. You can click on here to link to a document, one of the maths skills documents as well, to help you gain some confidence if you need it with working with standard form. Okie dokie. So we look at our starter of our question here, and we're told about bacteria at different temperatures. So independent variable, we're changing the temperature. We've got flask 1, 2, 3 inoculated with 1 cm cubed of broth at 20, 1 cm cubed of broth at 30, 1 cm cubed of broth with no bacteria acting as a control, and incubated at 30 degrees. Aseptic techniques used throughout. At set times, they removed samples and measured the number of bacteria. State one further variable the student should have controlled in their investigation in order to produce valid results. So valid refers to control variables, okay? Different to this flask 3, which is basically a negative control. This is to show that you wouldn't get any bacterial growth, without adding your, inoculating with your broth. And valid results refer to control variables between all three. So you need to think, you need to make sure that they're going to grow at the same rate. Ensure that they, et cetera, and so that the only difference that's going to affect the rate of growth is the temperature. 20 and 30. So you would need to make sure that you have the same sort of, volume, same starting population of bacteria, the same concentration of nutrients in the broth, the same pH. That's buffered. And we can see here, what else have we got? Oxygen concentration flask. I suppose I wouldn't have gone with that one. Number of bacteria in broth at the beginning. That's odd. Starting population, I would have thought they'd call it. Volume removed from each flask each time. Yeah, obviously. So if you were sampling and then counting, that would need to be the same. Volume of broth in flask. Actually, it doesn't talk about that. Oh, starting population. Here we go. Yeah, allow that. So, again, they'll often do that. Lead in with a sort of module 1 style question. Practical skills. Then we get onto some information here, and this is where it gets a bit more interesting. They use the following procedure to determine the number of viable bacteria. From each flask, 0.1 cm cubed was removed and mixed with 9.9. So you should be thinking, right, okay, so now I've got a sample of 10 cm cubed. This is tube A. Serial dilution then proceeded as shown in table 19. So we then took 1 cm cubed of tube A with 9 of water. Okay, so we've diluted by a factor of 10. So it's 1 in the total volume 10. Okay, so we've diluted it by 10, and again, and again, and again, and again. So we've actually diluted tube A 1, 2, 3, 4, 5. Yeah, so we've divided the number of bacteria by 10 each time. So we've actually divided by 10 to the minus 5. Okay, we've taken it down to. Then from each tube, 0.1 cm cubed was cultured. Okay, and then go to our next slide, and they give us this. So this is our 0.1 taken out of tube A, 0.1 taken out of B, 0.1 of C, and it was cultured. And this is sort of showing you full coverage. Okay, you expect the entire agar plate is covered in bacteria. Tube D, we start to see colonies. Okay, but there are too many to count. Tube E, you could count those, but then divided by a factor of 10 again, we get to tube F. Here we've got a few, colonies that we can clearly, easily count. Students used tube F to calculate the number of bacteria present in the original sample. Use tube F to calculate the number of viable bacteria present in the original 0.1 cm cubed sample from flask 2 after 7 hours of incubation. Give your answer in standard form. So if we go back, we need to find out what are we looking for? We're looking for the number of bacteria present in the 0.1 sample, okay, taken. So here we've got 2 0.1 samples. We've got this one cultured for 24 hours. We've got this one here, 0.1 was removed and mixed. So you've got to identify which one of those, okay, what was done first, and essentially, not done first. We're referring to this is the one, okay, that we are looking for. Okay, so we are looking for the amount of bacteria in this 0.1 cm cube taken from the flask. Okay, here we've got tubes, and in the question, it clearly says in the original 0.1 sample from flask 2. So this is where we've got to get to. We then can count here in tube F 1, 2, 3, 4, 5, 6. We have 6 colonies. We assume that each colony has been started by one single bacterium after culturing. So we would say that our sample contains 6 bacteria in this tube here. So we multiply that up by 10. 6 times 10, 60, 600, 6,000, 60,000. And then again, okay, to get up to tube A, 600,000. Okay, so we've gone up and times by 1, 2, 3, 4, 5. Yeah, so 6 times 10 to the 5 we've got to. That tells us how much was in tube A, but tube A was made up of 0.1 from the flask in 9.9. And this is where you'll often see the way the questions are designed. You might well get this far and then think, oh, okay, so it's another 10 to get to here. Okay, so therefore you go to 6 times 10 to the 6. However, it's not 10 here, is it? Because 0.1 out of 1, okay, is not the same as 1 out of 10. Yeah, this is essentially, if we move the decimal and make it 1 out of, that becomes 99, 100. So instead, it's actually 2 factors of 10. It's 10 times 10 times 10. So we end up going 6 times 10 times 10 times 10 times 10 times 10. So times 5 lots of 10, and then another 2 to get to our answer. So our answer ends up being, in standard form, 6 times 10 to the 7. Okay, so you can see here on the mark scheme, you can see here one answer if it's not standard form, and 1 mark for 6 times 10 to the 6, which is a common. I don't know how you'd get to 8, but maybe just miscounting or something. But I would say that it was designed to get people to get that answer. So again, not difficult if you know what's going on and you take your time and think about it. Easier said than done under exam conditions, I grant you, okay? But you've got to have confidence, and I hope that this will have helped.